Okay, so video 2 for prey location in barn owls is now going to look at the neural pathways for sound localization and discuss the Jeffress model of interoral timing differences. And then finally, complex receptive fields and how lateral inhibition helps to create those. So here's the neural pathway. You've got a simplified version from your book on the left, and then a more complex but also color-coded version on the right from a more recent paper. But essentially what you have in both is that information comes in from the cochlea on the 8th cranial nerve, and it enters the midbrain structures that include the nucleus angularis, the NA, the nucleus magnicellularis, the NM, and then those are going to feed information up further, up the chain further onto the cortex. And the pathways on the left are a little harder to see. That's why I included the one on the right. The green pathway, and that correlates with kind of the very complex boxes on the left-hand path or figure, is for interoral timing difference. And the red pathway is for interoral level difference. And so what you can see already is that there are some shared structures higher in the pathway, but lower in the pathway, that information is processed separately and independently. So for interoral timing differences, the first place in the brain and in the processing pathway that neurons start to respond to a particular interoral timing difference is in the nucleus laminaris. So that's where the star is indicated on both of those figures. So this is where you first get some specificity for a place in space that corresponds to a timing difference between the two ears. And so, of course, in order for that to occur in the brain, for the brain to be able to process that information, it needs to receive inputs from both ears. And the nucleus laminaris receives inputs from both ears, so it meets that qualification. And then it responds selectively to different interoral timing differences. The information for the ITD and ILD, the interoral level differences, converge in the inferior colliculus, which was also a structure that we talked a lot about with the bat. And the inferior colliculus, you have some pretty strong specificity for place, especially in the ICX. And then the information moves on to either the optic tectum, which is that red kind of semicircular shape, the OT, or goes up to the forebrain, to the cortex for higher level processing, and that happens via the thalamus. So we're going to be mostly focused on these midbrain structures, and it turned out in the, I mentioned the optic tectum, that's where you get overlap between both the visual map of space and the acoustic map of space. So when we talk about plasticity, we'll be talking about how the inferior colliculus sends information to the optic tectum. Okay, so here's the ICX, where you get a combination of ITD and ILD, the timing differences and the level differences, in order to create that elevational and azimuth map. So you get a 3D map in the ICX. So each neuron in that brain structure is responsive to a particular azimuth combined with a particular elevation. And that's going to correlate to a particular space in 3D space around the owl. And so you've essentially taken sound, timing, and level cues and turned them into a place signal in the brain, which is just very cool. So here's a neuron, and that black circle there represents what's called its receptive field. And so basically all sensory systems have receptive fields. So for touch, it would be like if you could take a circle or a pen and draw a circle on the back, say, to represent the receptive field of a particular neuron in the somatosensory cortex in the brain. If you were listening to that and you touched the circle, it would fire an action potential. If you touched outside of the circle, it would not. The circle would represent its receptive field. In other words, what is the sensory stimulus that causes it to fire an action potential? And for the ICX, that's going to be a sound coming from a particular place in three-dimensional space. So there's a circle there on this drawing that represents where, if a sound happened in that area of space around the owl, then this neuron that you had an electrode in and you were listening would fire action potentials. And you can see that the further you get away from its receptive field, the fewer action potentials that neuron will fire. So in thinking about intraoral timing differences and azimuth, we're going to return again to this concept of coincidence detection and delay lines, just like in bat echolocation and detecting the range. But in this case, instead of comparing the time delay between giving a pulse and the returning echo, now you're comparing the timing delay between when a sound hits one ear compared to the other. So in this drawing, you can see that the sound arrives from not directly in front of this person. And so it's going to hit the left ear slightly before it hits the right ear. And that is the intraoral timing difference. And of course, the more off-center the sound is, the larger the intraoral timing difference. And so our brains can make use of that intraoral timing difference to calculate how far off-center a particular sound is originating from. And in the 1940s, this guy, Jeffress, came up with a model that he proposed as being one way that neurons could translate intraoral timing differences into a place signal. And again, this was like the concept of delay lines and coincidence detection in those lines. And it turns out in the 70s that through barn owl research that this idea was in fact accurate. And so what we have in this figure, in the upper example, is a sound coming in with no intraoral time, with intraoral timing difference. So the sound is coming from the left side of the ear, of the head. And so it's reaching the left ear at time 0, but then it takes some time to travel to the right ear, and that time is now 30 microseconds later. And so the neurons are both converging onto a neuron in, we'll say, in the nucleus laminaris. And because those signals are 30 seconds apart, you're not getting any summation, and so you're getting a weak output. But if you incorporate a delay line that is exactly 30 microseconds long, then you slow down the signal coming from the left ear by 30 microseconds, and therefore the signals reach the nucleus laminaris at the exact same moment. You get summation, and you get more action potentials fired as a result. So that's the Jeffress model. It's very similar to what we were talking about in the MGB region for bat echolocation. This time we're in the nucleus laminaris, and we're comparing intraoral timing differences between the two ears. So we have coincidence detection and systematically longer delay lines from each ear that translates a time code into a place code. And so again, this is the same thing we saw before, where you have sounds coming from different regions of space. So let's say a sound is produced at E, then it's going to reach the right ear first. So here comes the right ear signal. And as that sound is moving to the left ear, we'll do that in green. The signal from the right ear is moving into the nucleus laminaris. And it's again, there's these delay lines, right? So it takes a little bit longer before it reaches B, a little bit longer before it reaches C. Meanwhile, that left ear sound is perhaps just arriving. So now finally you get the left ear is starting to transmit that sound some microseconds later. And it turns out that this delay line is going to be the point where those sounds actually coincidentally meet in the nucleus laminaris. And so neuron E is going to fire an action potential. And that action potential that comes out of E is now essentially equivalent to a place code. And what I mean by that is that it matters that neuron E is located right here in the nucleus laminaris and not right here or right here or right here or right here. It's a physical place, it's a map. It's now a map of space because E represents a space right there coming from the right side of the bird. Whereas all of those blue boxes I drew were sequentially moving that way. So you managed to turn the amount of time it took to reach both ears into an equivalent place in space that it means that sound must have occurred from. So if we look back at our neuron that had a receptive field in this picture here, so right here. So that's the receptive field. That's equivalent to a sound arriving at the right ear faster than the left. You can see that if you're the owl, that receptive field is to the right of the owl. And so that must correlate to somewhere around D or E. And you can see that that signal then comes out and you can start to build a sound map in the inferior colliculus, and then you can map that onto interaural level differences to get, so that's where this is ITD coming out of the nucleus laminaris. ILD is coming out of a different pathway, but they're converging in the ICX. And so you have both elevation and azimuth. But one problem that that all sensory receptors have is how to enhance the acuity of receptive fields. In other words, so like, again, here's a visual example because it's so easy to think about visual things for us. So if the dark bar there represented a receptive field of a neuron, so it could be visual, if you're talking about visual, or it could be an area in space where if you played a sound, then you would get a lot of action potentials. And that's surrounded by an area of sensory space that does not fire a lot of action potentials. You just get background noise, so a few action potentials here and there. That receptive field can be enhanced, or the edges can be defined by increasing contrast at those edges. So now, if you increase the contrast, you can visually see that bar much more easily than in that scenario. So in increasing contrast is important in sensory perception. And if you think about how can we do that in terms of a neuron's responsiveness, how can you create contrast? So we know that in the receptive field that neurons fire a lot of action potentials, and that's surrounded by the rest of the sensory space that fires just kind of weak background noise. So if we're looking at this visual example, you have excitatory is a darker shade. You have a kind of background level of noise that's this medium shade. So then how would you get the light bars that increase contrast? To do that, we would need to flank the excitatory receptive field with an inhibitory receptive field. In other words, if a sound were to occur just on the edges of the excitatory receptive field, it would actually inhibit the neuron. And so you would effectively get a clearer line of what is the receptive field for any given neuron. So here's an example of these kind of more complex receptive fields. So we have our receptive field. I tried to do some little animations. They're super simple. See how it goes. So if you look at this receptive field in the globe, so on the upper left, you can see I put a blue dot there in the positive region. And basically, if you move that receptive field, oh wait, I don't want to draw. If you move that receptive field or an electrode, say, sorry, you're listening to one neuron with an electrode. What you're moving is a sound in space. So you have a speaker producing a sound at that blue dot, which is in the middle of its excitatory receptive field. But then you move the speaker a little bit closer to center. And what happens to the response profile underneath of it, well, you can see that's a response profile. So you've got now you're mapping it in three-dimensional space, but the elevation of the graph is referring to how many action potentials it fires. So you can see the blue dot right now in that graph is at the top of a mountain of action potentials. And that's because you're right in the middle of its excitatory receptive field. But what happens as you move the sound out into the edges of its receptive field is that you get, wow, that happened really fast. It just didn't work out, did it? You get basically the ball drops as you move further out, further and further away from its excitatory center. And then it actually reaches a low, a valley, before, as you move further and further away, you return to kind of the background level of noise. This is an example of a more complex receptive field because you don't just have the center that's excitatory and then it just kind of goes to baseline levels. You have an area that surrounds it that is inhibitory. So that's that in this zoom representation, you can see the negative inhibitory area, where if you played a sound there, it would actually suppress sound or the responsiveness of the neuron. And this may not, basically where this becomes useful is on kind of a population level of neurons. So when you're listening across, say, the entire inferior colliculus, and you have one neuron that has the response profile that's right there, I'm going to draw a response profile of a neighbor neuron. So this is the next door neuron, and it has that kind of a response. Then if you get a sound, I'm gonna draw one more. If you get a sound that's here, you're gonna have action potentials coming out of this neuron, and you're gonna have background action potentials coming out of this neuron and background action potentials coming out of that neuron. If you have a sound that occurs here, then it's in the inhibitory area of both of those neurons. So you're going to have a response kind of like that. I'm going to run out of colors again. And if you have a sound that comes there, then you're no longer inhibiting that one. You get that. And so basically you're starting to get, if you do this with, thousands of neurons that all have closely related, overlapping, but different receptive fields, then you're going to start building a population code where that the brain can actually interpret better the exact location of that response. And I can, usually when we talk about, or a system that's commonly used for this is visual systems. They have the same kind of on center, off surround receptive fields. And so in the visual system, it's easy to then find lines of color change or contrast. And turns out that the same thing can happen in the acoustic kind of spatial mapping system. Okay, so how is this achieved? How do you have an inhibitory region of in receptive fields? And so for that, I just want to talk about a concept called lateral inhibition, um, because it can really increase the contrast between nearby receptive fields. And the concept is basically that an excitatory neuron sends its excitatory axon down the line, right, from B to B to B as you're in place, right? So you have your nucleus laminaris up to the inferior colliculus, and then to the optic tectum. And perhaps place B, the interoral timing difference that corresponds to place B is going to be preserved via a labeled line concept all the way up into to create a spatial map in the ICX. But those axons might also send inhibitory axons to their neighbors. And we're going to see how that enhances the acuity of a signal. So I'm going to start by demonstrating what happens without lateral inhibition. So we're going to say that that neuron A is weakly excited by an incoming interoral timing difference. So it's close, but it's not perfect. And same with C, but in the middle you have a perfect match. And so you get strong coincidence at coming out of B. Sorry, strong excitation coming out of B. So you get a lot of neurotransmitter out of B because it's the most excited. And then you get a little bit out of A and C because they're weakly excited. And so then you're going to have the same down the line, right? Weak or strong excitation. And so out of the nucleus laminaris, here, this graph, we can graph how many action potentials come out of those neurons. And what we can see is that we have a weak signal coming out of A, A, B, C here. A stronger signal coming out of B, and then a weak signal coming out of C. So this is just the number of action potentials that those neurons are firing. And now I'll draw what happens with lateral inhibition in a different color. So we have these signals coming in from the nucleus magnocellularis. B is more excited, A and C are weakly excited. But what the nucleus laminaris might do is send inhibitory projections to neighboring neurons. Okay, so going down the line there. So you can imagine that they have their excitatory axon, but now the axon is going to branch and also send an action potential to an inhibitory synapse. So I've drawn this straight line here to represent that this synapse releases GABA instead of glutamate. Okay, excitatory synapses release glutamate. Okay, so if you think about this strong excitation of B, it's going to both release a lot of excitatory glutamate onto onto the next cell in the B labeled line, but it's also going to release a lot of inhibitory GABA at its inhibitory synapse on its neighbor's synapse. Okay, so you can see that this is presynaptic inhibition. So what it's essentially trying to do is keep A from releasing glutamate. And so a lot of GABA release will prevent the neuron from releasing glutamate. Because the glutamate causes chloride influx, which is going to prevent the calcium channels from opening, so you don't get any glutamate release. Keep in mind that the weak signals traveling down the A and the C labeled lines are going to also release some GABA because they have a few action potentials happening, but it's not going to be enough to counteract the frequent and higher, kind of stimulation of the B labeled line that's being strongly activated. And so the net output then is that you have low or no excitation of these weakly stimulated labeled lines, whereas you still have excitation coming out of B. And so you have enhanced the difference between what the ICC is seeing from its connections from the nuclear laminaris. So instead of getting a little bit of excitation of A and C and a lot of excitation from B, you get essentially no excitation from A and C and a lot of excitation from B. And this is one way through lateral inhibition that nervous systems can enhance or increase the accuracy of signals coming in. So if we look at this of our kind of labeled lines in the nucleus laminaris on the left there, and you get a particular ITD that results in an output code, that's going to travel to the ICC and meet with ILD information in the ICX, and you get your specific receptive field. And then, of course, you have these regions of inhibition flanking that excitatory receptive field. One problem with interoral timing differences is if you get changes to how sound is reaching the two ears. This can happen in adulthood with damage to the pinna in the outer ear, but it happens more consistently in every vertebrate animal that develops as an organism grows from being young to being an adult. So on the left-hand side is a change in head circumference for human female babies. And you can see that there's a large change in the circumference during the first 5 years, and it's even still continuing even at a much smaller rate after that. And so if the difference in the distance between the two ears changes, then the interoral timing difference is going to change as it relates to acoustic space. And the same is true in young developing owls. The space between the two ears changes as the owl grows. So you need to be able to adjust how these neurons connect, because as the space between the two ears gets larger, it may be more likely that this neuron should connect here, and this one should connect there, and this one should connect there, and this one should connect there. In other words, you need to have a way to adapt and change the connectivity in the nucleus laminaris if you want to maintain the link between the interoral timing difference and the actual acoustic space around the animal. So we're going to move into neuroplasticity, and I just want to reiterate this again, that the reason that owls are so great for studying plasticity is that they have sound and visual processing centers that communicate closely together. And so when you change visual or auditory perception, you can get a misalignment. This happens during growth. By studying owls, we've understood some of the mechanisms for how plasticity can help compensate for this. Okay, so we're going to stop there, and we'll start the next video with neuroplasticity.